Welcome back. Let's kind of start where we kind of left off. We had talked about cell cultures, facts, separating cells. We talked about the use of antibodies, monoclonal, polyclonal. Okay. We figured out a way to separate fractions of cells, so cell by centrifugation. Okay, centrifuge, we rate zonal, isopycnic, all that. We could get fractions, and each fraction could contain an organelle, and that led to protein, right? Proteins, okay, proteins. And along the way, we could do from centrifugation cell-free systems. We could then mimic what was going on inside cells. So a cell-free system here. Okay, cell-free. And then for these proteins, clearly if we had found them in an organelle, they must have some function, right? So we're very interested in function of proteins because that leads to functioning in the organelle, and of course organelles are in the cell, and there must be connections between organelles, cell physiology. And when we purified, say, mitochondria, if you're looking for proteins in mitochondria, well, you purify it, and if you find your protein there, that gives you a location. And a location should then lead to questions about, well, this protein's here, what does it do? What's it functioning? And again, having location probably eliminates a lot of questions. In other words, if it's not in the Golgi, then it probably doesn't do Golgi functioning. For instance, if it's in the mitochondria, it probably does. And so the next thing, we want to continue studying proteins. And there are methods to look at protein interactions and protein blotting, and just using various techniques and tools to study multi-protein complexes that gives you this idea of function here. So there's tech that's been developed in order to further look at protein function here. So this next block of material, at least for a while, we'll be still dealing with techniques, but we're past this point and we're now into looking at specific protein function localization. And so let's talk a little bit about protein blotting. The word blotting means taking one piece of, say, nylon or nitrocellulose and taking a sample protein, messenger RNA, DNA, and transferring that from a acrylamide gel or an agarose gel onto then the blotting paper, which then can be further manipulated. So this is a cartoon sketch showing protein blotting, not all the blotting, but just three main types. A southern blot, a guy named Southern, 1975, used a blotting to detect pieces of DNA. A northern blot is, there was no Dr. Northern, it was just a play on Southern, was for mainly messenger RNA, not just RNA, messenger. And then probably in cell biology, we do a lot of Western blotting here, and that's dealing with proteins. So protein blotting, Western blots are the world we live in here, trying to figure out function and location. These DNA and messenger RNA will be more in the cell biology course I teach. And if you want the extreme here, this is the blotting compass, And some of this I don't even get into, but just if you run across in the literature, consider this a reference guide, but there are lots of different variation in blotting techniques, RNA, proteins, DNA, proteins, protein, DNA, lipids, modifications, RNA to DNA, lipid to some type of PTM factors, all sorts. We're going to just focus right now on Western blotting. And a guy named Tobin developed this with Burnett, but Tobin, he actually developed the transfer method, and we call the buffer or transfer buffer, we use Tobin buffer after Dr. Tobin. So this is a typical uh, breakdown, kind of a deconstruction of a Western blot here. And that, number one, that gel is a polyacrylamide gel. So that can be SDS Page, SDS, remember our friend SDS Page, we actually talked about a 1D and 2D electrophoresis. So this gel could be 1D or 2D electrophoresis here. 1D or 2D gel. This is just showing how you cast the gel. Here's the gel comb. Here's the little holes here. Gel knife, I don't even know what that's for. I never used the gel knife. And then here's the chamber. You put this in, you apply electrical current in electrophoretic buffer, and proteins, if they're SDS charged, they'll move to the positive pole, which will be the platinum lead down at the bottom here. And so after you get the blot here, you do a sandwich in a cassette, and these are looks like little waffle pieces of plastic here. And there's our gel that we just electrophoresed, and there's some blotting paper in white. And then right on top of the gel is nitrocellulose, some type of nylon, or other types of membrane material that's cut into the same size as this gel slab here. Okay, and so there's some sponges. They soak up the Tobin buffer, and this cassette is locked down. You kind of make a layers here. You lock that down, and you get rid of all the bubbles, and then you apply that. And I don't, okay, so they don't show you that, but you apply that cassette to another electrophoretic chamber, and the proteins move out of the gel and they're electrophoresed into the blot itself. So here's a cell with DNA, RNA, and protein, and I just wanted to show you that we have DNA and RNA here, messenger RNA, but we're protein. And here is the polyacrylamide gel. We blot it, and it says transfer separated sample to membrane. So in this slab, all these proteins, okay, little bands, or if they're 2D, little spots, okay, little spots, protein spots. Either way, the electrophoresis cassette that we built, you apply electricity, and you're making, because remember the proteins are still charged, and we have another set, and of course the plates will have to be, instead of the vertical, it will be the horizontal, and the protein moves across and hits the nylon membrane. So go back to that figure before, and those bands or spots end up in the same location that you separated them, and that's the key, same location. So the bigger large mass proteins will be in the top, the smaller proteins at the bottom, and so forth, right? And the spots as well in the 2D. So now we have a blot that we can then do the next part of the Western blotting. So we've already blotted it. So now we're doing the Western blot, and we have to have some type of primary antibody to some protein. If we're interested, what's this spot? And we have the secondary antibody that has to detect and amplify the signal. And here is, and these can be different samples. Say that's cancer and wild type, and is the protein in one but not the other, and contains specific protein. So we add the antibody and it sticks to the protein. The secondary sticks to the primary antibody. So we have the goat antibody, then we have the rat antibody, then goat anti-rat secondary antibody, and we do some type of signal generation, and we're able to see. And these are pre-stained standards, and we can develop a standard curve here and predict what molecular weight those are. And you can measure this intensity of this. So how much protein is in this cell line versus that, or in that cell fractionation sample, or in that organelle fraction, right? Or this is mitochondria, this is nucleus. Okay, is the protein in both places, or in one or the other? Okay, Western blot allows you to look at that. And here's just a little bit deeper protein blotting. Here's the blotting paper. And here is the just showing the membrane here, and then notice that the proteins will is negative charge will move to the positive pole. There they are. And once we get the blotting that occurs, it's hours. It takes hours. You take that membrane and you block it, then you add your primary, you add your secondary, you add some type of substrate if this is an enzyme, and you get a enzymatic color reaction at the point where the protein is that this antibody recognizes. So there are companies that will sell you antibodies to key proteins. So you could scan any set of proteins, any set of cell samples with any antibodies you want. Or if you've got a new protein, then if you made a new antibody, you could test it on the blot there. And so here's the transfer. You acrylate everything, transfer, keep the temperature, everything's nice. Here's our cassette sandwich. This is the electrophoretic chamber. And so what happens is the platinum wire is on over here, okay, on the cell wall, on the sides of the tank in that direction. So I don't know which way it's moving, but oh, here it is. Direction of transfer. Remember, in electrophoresis, the direction was down. Here it's across, okay? And so you get the anode platinum wire over here, and the cathode is there. Proteins have a negative charge if they're SDS stained or applied, and proteins move out of the gel because it's just a porous kind of a, remember, an acrylamide is kind of has pores, and the proteins leave those pores and hit that membrane. That membrane cannot transfer membrane. That membrane cannot be porous that proteins would go past it, because proteins would. They'll go directly to the platinum wires. So if you didn't have a transfer membrane, the proteins would, if you forgot to add it, the proteins will leave the gel, go through the filter paper, the blotting paper, the pads, and hit the platinum wire through the support grid, and that wouldn't do you much good. So how do we know if the transfer is working? And remember, you had the primary, the secondary, you block it, you had primary, secondary, and then you have an enzymatic reaction. Well, fortunately, companies, here's Thermo Scientific Page Ruler Plus, they actually did a trademark, and it comes with proteins that are in a mix that you add to a well, and they're already pre-stained with chromatic chemicals. And these proteins are 9 proteins, blue, orange, and green stain proteins from 10 kilodaltons to 250 kilodaltons. So it's very small to very large. And so when you electrophoresis, that the standard should separate as shown in the document here, or in their, in their company thing here. And so when you blot it, these should go and be on the blot. So here's the blot. So so you're saying, well, if I put it in opposite, well, then it won't be on the blot. Okay, it would be in the buffer. Okay, that'd be bad. Everybody would get cranky. And so here's a description of this product, and it talks about, looks like the reds are at 70, 25, and looks like they've got do they have a gold or an orange dye there? Blue, orange, or green. So maybe that's what they're calling green, and then the rest of these are blue dye. And so they're able to determine that the transfer worked, determination 1, and separation worked, determination 2. Okay, and you can measure with a ruler the distance in millimeters. And now you can plot on a graph paper this molecular weight versus the distance traveled. And if you plot that, it should be a straight line. Okay, straight line. That's not straight, but straight line. And then if you have an unknown, say a protein right here, then you measure how far the protein went, and then you find the line and drop down, and that'll be size of protein versus distance here, the X and Y axis here. And so here's just another picture of the cassette, the sandwich. This guy's got some forceps. Here's some pre-stage standards, or that he's just showing that it worked there. So, okay, so that's him putting it on the gel and then after transfer. Okay, here's the transfer apparatus. You can do a dry transfer. It tends to run for hours. The submerged tends to run overnight, depends on the electrodes. Obviously, the plate electrodes probably cost more than the wire electrodes, because you have to have more platinum to cover the whole plate versus some thin wires. So anyway, this is, again, about the third way I've shown the transfer. So things that could go wrong: you forget to add protein to the sample. That's not good. If you had bubbles between the gel and the membrane, then the bubbles will interfere with transfer. So you take a little, usually we'd take a glass pasteur pipette and kind of roll it like a roller, biscuit roller, and roll across and get all the bubbles out. Then we would stack this cassette and put it in the transfer chamber there. So Western blot finds potential target proteins we're interested in. We can figure out, if we don't know their molecular weight, how much protein is there. So the amounts of different proteins. If there's a virus or a bacterial protein, we can detect pathogen in a patient's blood or lymph or spinal fluid or nasal secretions or biopsy, and other kinds of cancer biomarkers. We can use Westerns here. Under certain conditions, we can look at proteins that will be highly or lowly expressed, and we can compare a disease, a stress, drug treatment, and see how the protein expression changes over time. And that can be key for understanding, say, a disease, okay, or a drug treatment. Here's just, looks like this company sells us a GenScript one-step Western detection, and they're claiming this is an hour versus traditional 4.5 hours, like we've showed here. And again, remember, the enzyme here is attached to the secondary antibody We have some type of substrate, and we have some type of product made. So this is the product, and it usually has some type of color, and we get some type of band or spot that has a blue or black, some type of purple precipitant, and that is said to be the protein there. Here's just another figure showing the protein, primary, secondary detector here. This could be an isotope if you want exposed to X-ray film, but there are different methods of detection in Western blotting. And again, as many antibodies as you have, you can do many lanes and detect what proteins are there that way. Detection of the protein can be colorimetric, could be radioactive. We could have a fluorescently labeled second antibody, and it could be chemoluminescent, that the reaction actually causes luciferase to be released, and that is detected by X-ray film. Luciferase is what makes fireflies yellow in color, and they actually found the luciferase gene and labeled that onto the secondary antibody or the luciferase enzyme. Okay, and so now we thank lightning bugs for helping us. You're saying, why would you do that? Sensitive, very sensitive actually, and you don't have to play with radiation. Okay, so the colorimetric's the cheapest, but not as sensitive. So you have a protein that's very low expressed, you probably need to do something very sensitive. So there's a sensitivity issue here as well. This is just another figure pretty much showing the same things, just different way to look at it. I like to show you different things. And here's 3 different samples or cell lines or fractions or organelles, and it looks like the proteins in all of them. Okay. And here's another one showing the primary secondary antibody here. So that is protein blotting. So again, go back to the questions. What are we looking for? Why are we using the blot? What can we determine from the blot? Okay. Sound like a test question? Okay, you need to know that. If I ask you, what's a Western blot? Why do you do one? Now you know. So anyway, I'll stop recording here and we'll pick up with the next topic. Come back real soon. Okay, let me stop.